Dr. Rebecca Ross, welcome on our SOPHIA studio. And I would like to confirm a few information about you and your presence on this deep sea symposium here at Aveiro. You come from the UK, from Plymouth University, and you have been putting a talk here on in the symposium about larvae, comparing, predicting the dispersal of larvae of deep sea organisms. What are in fact those larvae? We associate larvae with some nasty little animals. We have flies and decaying matter, and so what's larvae in the deep sea, and what's the dispersion of those larvae, please?
Okay, well, larvae actually are very similar to what you're talking about, these little disgusting things for insects, but it's the same thing in the deep sea. They're the juvenile stage of the animal. So in the deep sea, we're talking about animals that, as adults, are attached to the seafloor, and their juvenile stages get released into the currents, and they're just absolutely tiny and carried very far by the currents. But the adult stage could be a coral or a sponge or something like that. Big, beautiful animals, and they have these tiny little larval stages. And the dispersal process is the ability of that animal to get away from where it was born. So the point of dispersing is really to try and avoid any kind of mixing of genes with your kinfolk, to try and avoid competing with your family, and to try and find more favourable conditions perhaps, or to escape really bad conditions. So every animal performs some kind of dispersal. We do as well. We probably move away from home. And it's common across all animals, but in the deep sea, especially with these benthic sessile animals, these adults who live just fixed to the seafloor, it is only the larvae who are able to make this dispersal process.
Yeah, it's amazing, because usually we do not think about that. We think of larvae as large pumpkin. We think about big sponges or so as some kind of large pumpkin that is planted in the seabed.
Exactly.
And while they have that possibility once in their lives of traveling wide over large distance. And it is important, obviously, to know how do they behave in this stage, and also what we can do in order to allow the process to flow naturally. Saying in other words, what we should not do to disturb their natural cycle of life. Have been any problems identified that could associate to our activities? I know you have been sharing a session on this, either from your work or from your colleagues in the session. How is mankind interfering with the dispersion of larvae?
How is mankind interfering? Okay, well, things like just the destruction of the adults. If we perform any kind of destructive seabed process that will wipe away the adults, then obviously they can't release their larvae and pass it on. And the problem there is also, if you don't protect areas where the adults live, even if they can release their larvae from a protected area, if they can't get somewhere where they will survive, then it is hopeless for them. So the point of putting something like marine protected area networks in is to be able to have these sort of islands where they will be safe and the larvae will be able to join between them.
Okay, so we are very much aware, both of us, that marine protected areas are something that may help, probably is necessary in some areas, but it is not enough in itself. We cannot just make small zoos for deep sea animals and then destroy everything around, because it simply will not work like that in many cases. And what do you think, at what stage are we now? We need to know much more about larvae. We are about knowing all that is needed, how we can put the scientific knowledge level now?
Well, we know very, very little, especially in the deep sea, about the larvae. There are a few studies that have been carried out with very few species, and they're really great work, but it's very hard to find out anything about the larvae. I mean, think about how difficult it is to do any kind of sampling down kilometers down in the water, and then to find these tiny microscopic animals and learn anything about them. So there's so much we need to learn. But in the meantime, we can do things like these modeling practices, try and come up with probabilities in order to try and figure out what could be the case, and then maybe we can backfill and cross-compare with other studies.
Yeah, in a certain way, you are doing some kind of inner space exploration and planning the orbits and so that you can have a good gathering of information and avoid costly mistakes and sending costly missions to areas that are of low probability.
Exactly.
Okay, I see.
Yeah.
And the knowledge we have about these larvae is widespread across the world's ocean, or is it concentrated in some areas that have been much more studied than others? What's your perception on this?
There are definitely areas with much more study than others, and of course there are countries that have better funding and are able to undertake these things. But luckily some species are in multiple areas, so research in one area will help people in other areas, but there is so very little learned about any of the larvae of these animals that there is so much work that we can do to try and help with this.
Yeah. So basically, we can say that we are still very much in the dark.
We are still very much in the dark. Absolutely. There are very few things that we can do, but there are any kind of population genetic studies. Those kind of things can help try and work out how related populations are to each other. You can perform some kind of geochemical tagging situation. So you can artificially put something in the water that will be recorded in the bodies of animals that have some kind of carbonate structure in their skeleton. So something like maybe snails, gastropods. They would be able to record the signature of that in that area, and then you might be able to find them again in another area, and you'll be able to see how far they have gone that way. Yeah. But then there's the models are what I work with the most, and they are really good for trying to simulate what is possible as a dispersal from a particular location. But they still need to be validated, and all these processes really need to complement each other, because none of them actually give you a reliable answer on their own, really.
Okay, so I wish you the best of luck with your work, and we have again here a case for raising cooperation between scientists in these deep sea areas. So thank you again.
Absolutely.
And it was a pleasure.
Thank you.
Thank you.
I think this is a great idea.
Thank you.